Yes, so thank you, Sophie, for such a lovely introduction. As Sophie said, my name is Victoria. I am a postdoctoral researcher at the Toddlerlab at Birkbeck here in central London, where we are a little bit sweltering, but I'll try to make it through with some high energy levels for this talk. I'm really excited to have been invited to talk here today. From what I understand, you've had a great lineup of speakers, and I'm hoping to add a little bit of a different perspective probably to what you've heard by talking about my work in developmental science. So trying to understand how we can use hyperscanning to investigate the development of collaborative skills, particularly in the preschool years, using wearable fNIRS. So my background is in developmental psychology. So I was trained in kind of traditional neuroimaging methods and psychological paradigms. Hyperscanning is a bit of a new world to me. And my initial interest in research was about trying to understand how children develop social cognitive skills. So how do they learn to represent other people's behaviors, their mental states, but also incorporate them with their own in favor of accomplishing particular social goals? And most of my work before joining the Toddlerlab officially was kind of looked like this. So we often would ask parents to come into the lab with their kids between the ages of 4 and 24 months, so quite young. And we would ask them to sit in this dark room and look at an eye tracker screen like you can see in this picture. So this is a Tobii eye tracker. And we would play different videos and show different pictures while we tried to get the child to look at the screen. We made everything as boring as we could in the room so that they wouldn't get distracted. And we would present this kind of series of pretty artificial, very often, stimuli that we also controlled. So we decided how long we would play something for, how loud it would be, what it would sound like, and measured how children perceived and responded to these kind of social stimuli that they were being presented with. But of course, this isn't really the real world. This kind of setup can tell us quite a lot about specific processes of perceiving social cues, but it can't necessarily tell us much about how social cognition develops in the real world, which we know is kind of really critical to understanding a person's developing ability to interact with another person. So we know that children learn during interactions with others. There's decades of evidence to suggest that the pattern, the dynamic of a child's interaction with another person really informs and is related to how they develop in lots of different domains. And I'm not talking about hyperscanning studies here, but just kind of looking at parent-child interaction using behavioral methods and EEG on a single participant and lots of different things, over the years. And we know that this is really important to understand from a hyperscanning perspective, because it's very likely that when a person is interacting with another person, that they're engaged in different cognitive processes than if they're just passively receiving social cues like they were in my postgraduate research here. So if I'm watching something as someone's talking, for example, I might be representing what they're saying, their behaviors, I might be predicting what's coming next. But if I'm interacting, I'm also thinking about how to align my own behavior with that other person. So to fully understand social cognition and the way it develops over the course of a human's life, we need to understand how children engage in this kind of dynamic back and forth, reciprocal exchange of information with others in what's often very messy kind of real life scenarios with their parents, with their siblings and friends, and later on with their teachers and their peers. So that's all well and good. I think it's an admirable goal to say we need to understand this stuff, but there is, of course, many methodological challenges. So all interactions, but especially children's interactions, are often very messy. This is a participant we had a few weeks ago, having a great time. We were lucky he wasn't wearing a cap at this point in time, but he's in the middle of a task and kind of decides he wants to do something else. So this is what I'm hoping to highlight today. That's probably a bit different from what you might be used to seeing. We very often study kind kind of interactions and hyperscanning between two adults with two fully mature kind of social cognitive systems who will sit and listen to our direction and be still and kind of click buttons. And that's very much not what we're talking about here. So we need methods that children will tolerate in order to do hyperscanning with children. So obviously we need things that they'll wear for long enough that we can collect enough data for our studies. But we also need equipment that will tolerate children. So it's not just a one-way street. It's really about having something that is robust enough that allows us to collect high quality data with good signal to noise ratio, despite all of the noise that children might choose to introduce to a particular setting. And of course, if we want to understand kind of naturalistic interaction, we need equipment that we can use in naturalistic paradigms, where we're not necessarily controlling exactly what's happening at every second, and that can be used in hyperscanning designs. So can be used to collect simultaneous measurement from multiple people, which I know is very familiar to you all here today. Probably don't need to redefine hyperscanning. So very kind of early days of hyperscanning literature involved using fMRI to collect neural imaging from multiple people, two people simultaneously. So this is an image that kind of is the picture perfect view of what an fMRI hyperscanning study might look like, where we have two people lying down very still in either two different coils or one machine with a dual coil. And we can kind of connect them in those machines and say, okay, you can talk to each other, maybe you can take turns on a task, or you can work together on a sort of limited task. And we know, particularly from this lovely recent review by Hakim and colleagues, that in the early days of hyperscanning in sort of the early 2000s, on the left-hand side here, you can see the little silver bars. There wasn't much out there for hyperscanning research, but what was available was primarily in the fMRI modality. And then around 2015, you see this explosion of this orange bar, which is representing fNIRS hyperscanning studies. So particularly in the last 10 years or so, we've seen kind of an explosion of use of this equipment. And particularly in our case, I'll be talking today about the Bright MK2 systems. This might be familiar to lots of you. And the benefit of these for us is that they are fully wearable, of course. So you can see on the left-hand side here, we have a child wearing a cap, and you can kind of see that the wire going from the optodes to the pack is actually just popped right in that Mickey Mouse backpack. So with this setup, that child is free to run around as much as they would like. Sometimes we also put the pack on the back of the cap. It depends on the size of the child's head, mostly, what they'll tolerate. And this allows us to do some really creative things in our hyperscanning. So for example, on the right-hand side, you can see two brothers who participated in one of the studies I'll talk about today. We tell all the kids, you're going to do this astronaut mission where you go to space and you need to wear this really cool cap that allows you to blast off to do your mission. And so they had just finished running around the table, which was how they powered their rocket ship, and then they posed for the photo. So they were able to do all of that while they were wearing the equipment. Just a very brief mention of how fNIRS operates. I'm sure this is very familiar to you all here today. Of course, with fNIRS, we rely on a phenomenon called neurovascular coupling, which suggests that neuronal excitation triggers blood vessel dilation, which causes an increase in cerebral blood flow to active regions. And this response is generally characterized by an overall increase in hemoglobin to active areas, particularly an increase in oxygenated hemoglobin and a sort of subsequent decrease in the concentration of deoxygenated hemoglobin. So we measure these changes in concentration using near-infrared light, of course. So we shine different amounts of near-infrared light in through the source, which you can see here in the middle of the two detectors, through the extracerebral layers and into the cortex before measuring how much light is either scattered or absorbed using these detectors on either side. From this, we can then estimate concentration changes in oxy and deoxy, which provide us with indirect measures from which we can make inferences about likely brain activity. And very often with fNIRS, we make a set of assumptions about what our signal represents. So we do all of that, we pop the caps on, we adjust all of our lights, we measure our signal, and then we say this signal that we've captured is representing functional brain activity, particularly of whatever it is we've evoked. So whatever task it is we're asking people to do. And we assume that this signal is completely representative of cerebral hemodynamics. So this means that it's coming from the brain, right, which is a big assumption, and that it's related by the property of neurovascular coupling to neuronal excitation. And this is the sort of set of assumptions we often make here on the left. But the reality is a bit different. So that's represented here on the right. You can see there's much more red kind of spread out in this 3D space. So the reality of any hemodynamic-based technique is that we are also measuring signal that isn't from the brain. So from the extracerebral layer, layers of the scalp, we know that there are changes in hemodynamics in the scalp. We often don't really know what to do about that. So we just sort of pretend it's not there, but actually there are ways we can kind of pull that information out and control for it and try to sort of clean up what we assume is our cerebral signal. We also know that particularly the extracerebral layer, but also cerebral hemodynamics are related to systemic physiology or contaminated by changes in systemic physiology. What I mean by that are things like heart rate and respiration, breathing rate. Of course, these things might also be interesting to us scientifically, but often they're not, and we still sort of have traces of those systemic components in our NIRS signal when we don't really want them there. And so I'll talk a bit today about how we approach this issue and how we tried to optimize our analytical pipeline to account for it. And it's particularly important in the context of naturalistic hyperscanning because we know that sort of this reality over on the right-hand side is present in our computer-based fNIRS tasks. There's research with adults that shows that actually, even when you're just sitting at a computer pressing buttons, we see contamination from systemic physiology. So we would expect it to be much worse when children might be jumping, running, moving around. And so it's particularly important in this context to account for that. Of course, I'm sure all of us here are already on board with this, but there are many pros to using fNIRS, which is why it's so well suited for our work. They are, of course, the bright MK2s are wearable. They're suitable for naturalistic designs and paradigms and settings, so we can have them not just in kind of dark rooms, but we could even wear them outside or out in kind of real world scenarios. And they are very child-friendly. So we've definitely found that in our work, and I'll explain sort of how that's looked for us. Because they're so kind of well suited to naturalistic designs and interactive paradigms, using fNIRS, lots of research with adults has shown that during interaction, the functional brain activity of often two participants will synchronize. So I'm sure all of you know of the concept of neural synchrony, which is just the temporal alignment of brain activity between two interacting people. Synchrony, of course, can mean lots of different things, and I think it's really important that we always be discerning about the modality we're talking about. So if we're measuring with fNIRS, it's very different to EEG if we talk about synchrony, and often that's not very clearly stated. So for the purposes of this talk, I'm talking about neural synchrony or coherence as the alignment of changes in concentration of oxy and deoxy in the cortex of person 1 and person 2. And this image is from a recent-ish review of adult hyperscanning literature using fNIRS during collaborative tasks. So you can see all the tasks here on the bottom pane. There's joint finger tapping tasks, joint drawing, math, joint singing, all kinds of different things that you can ask adults to do. During all of these collaborative tasks, participants' brain activity seems to synchronize in frontal regions responsible for executive control and attention, error monitoring, kind of sequential planning of steps towards an overarching goal. And also in temporal regions, which I'm sure, as we all know, are thought to be involved in social cognition, so mentalizing, judging other people's responses, understanding what they might be doing. And you also see some evidence in lower temporal regions here involved in knowledge retrieval. But of course, none of these tasks are possible when you're first born, right? So collaboration is not a skill that we're born with. It's something that we learn. And particularly, it seems to be that collaborative skills develop from 3 to 5 years of age. So when you ask 3-year-olds to collaborate, they often will grab pieces or grab bits of whatever it is, and they'll work separately next to each other. But when you ask a 5-year-old or 2 5-year-olds, they're much more able to engage in that collaborative process. So investigating kind of how collaboration develops is it's a really good time period to look at that in the 3 to 5 year kind of preschool age. So today I want to talk about a sort of set of studies that I've been conducting in collaboration with Denis Marichal and Paola Pinti, funded by the Leverhulme Trust. We've been really lucky to have 4 years of funding to set up the Toddlerlab's first hyperscanning research. And in our first study, we just wanted to see if we could replicate the common finding that I just showed in adults, that neural synchrony emerges during collaborative tasks more so than individual tasks. And in particular, there was a well-cited, very popular study by Wynn and colleagues in 2020 that looked at this in 5 to 6-year-olds and their mothers. So we just wanted to replicate what they found, really, and see whether we could reliably replicate their results. So you can see that they found that coherence between the child and the parent was strongest in cooperation, cooperative problem solving, than individual problem solving, and also then in rest. But we also wanted to add a few extra pieces. So we wanted to make sure that our wearable Bright MK2s didn't throw any curveballs into that equation for us, that we were able to set it up in a way that allowed us to replicate the results with wearables. But also we wanted to optimize our pipeline a little bit to reduce physiological noise, the issue I talked about earlier, and also to add a bit of a computational step to test whether synchrony is entirely explained by a common task. This is something lots of people try to do, and we took a bit of a different approach to see whether that were really the only explanation. We also wanted to test the role of information that might be available on the other person's face. So there's been lots of evidence in adults that eye gaze might be related to neural synchrony. These are some plots from Joy Hirsch's lab at Yale from 2017. And they found between 2 adults that eye-to-eye contact, this is showing deoxy in their results, eye-to-eye contact was related to stronger synchrony in temporal regions than eye-to-picture contact. And that was replicated by Adam Noah in 2020 with video stimuli as well. But then I know there's also been a talk, I think, by Ira Marriott-Hairsine, who's found maybe things are a bit more nuanced or complicated than just, eye gaze means greater synchrony or not. So there's a bit of a question mark, particularly about what this looks like in children and their parents. So our first study was published a few months ago. You can find much more detail about all of it there. But we asked about 47 pairs of 4 to 6-year-olds and mothers to come to our preschool lab, which, as I said, is in central London, kind of by King's Cross. And it looks like this picture in the top right here. So you can see it's designed to look like a preschool, but with the addition of lots of cameras, I would say. And we had about, I think it's 13 additional dyads come in who were excluded for different reasons. And our final sample was 47. Our task was replicating Wynn's task from 2020. You can see that in the top picture here. So we gave participants these templates and asked them, and the pieces of the tangram puzzle, and we asked them to make the pictures either together or separately. We had 3 conditions. So we had full collaboration where they just sat down at this table and they worked together like they would normally. And then we had fully individual, where we drew a screen between them, asked them not to talk to each other, and they worked separately on different puzzles. So that was our individual problem-solving condition. But we also had a third, which was collaboration with screen, or partial collaboration, where they were given 1 template and 1 puzzle, and they were asked to work together to build the picture, but they weren't able to see each other's faces. And this was to address that question that I mentioned about the availability of face information as a potential necessity for the emergence of neural synchrony. We had 3 trials, each 2 minutes long per condition, and they were separated by 80-second rests at the beginning and end as well. And both the children and the parents wore the bright MK2s that I referenced. Our array covered the temporal parietal junction on both sides and also bilateral prefrontal cortices, which are regions thought to support mentalizing and planning and execution of kind of steps to reach a common goal. We had 18 total channels, and again, there's lots more detail about our array in the paper, but we had 16 long separation channels and 2 short separation channels. The short separation channels were placed in one in the left PFC and then one in the right TPJ to kind of cover that cortex as much as we could. They also wore pupil core head-mounted eye trackers, which I'll discuss in our sort of second study that we're working on at the moment. You can kind of see that the parent over her right eyebrow, she has a small camera. That is a kind of worldview camera that shows us what she's seeing in her visual field. And then those little arms by her cheek are the pupil detectors, which allow us to estimate her fixation in her visual field. And I'll show a video of just what kind of typical collaboration between the parent and child looked like. So I think the mother here is kind of showing the child where to put the green piece on the cat's face. So pretty kind of typical collaborative interaction between the parent and the mother. We pre-processed our data in MATLAB using Homer 2 functions. And as is typical, we first just explored the raw intensity signal to make sure that we had lots of channels with high signal to noise ratio. So anything that had low signal to noise ratio, we were able to exclude. We then corrected for motion artifacts using a wavelet-based method, which we found at the Toddlerlab in general in any naturalistic fNIRS study has worked really well for us, not just in hyperscanning designs, but any study where we have a child who's running around and moving quite a lot. And we used the parameters that we found work well for children in particular of this age. Then we applied a bandpass filter and converted our optical density to changes in concentration of oxy and deoxy using age-corrected parameters for the children and the mothers again, of course. As most fNIRS hyperscanning research does, we used wavelet transform coherence as our calculation of neural synchrony between the parents and children using the traditional toolbox here. Anyone who's interested in kind of the specifics of that pipeline, it's publicly available on my GitHub, so you're welcome to go take a look at it. And this is what it looked like for anyone who's unfamiliar. So this is an example of a spectrogram between channel 1 of the mum and channel 1 of a child. And what we get out of a wavelet transform coherence calculation is just a correlation across time and frequency between mum and child's brain activity in that channel pair. We can then average across our frequency band of interest in the purple there. So we might imagine that these blobs are kind of episodes or periods of high coherence between the two. But if we average across that purple band, we then get kind of one average value in our frequency band of interest over experimental time. And then we can also average across our conditions. So our trials for collaboration, for example, we might get one value per condition, and then we might also decide to average across all of our trials. We didn't do this in this paper, but often if there's students working on this data, or if you just want to sort of deal with less data, sometimes that's a useful thing to do. So of course, as I referenced, often we would just take our long separation signal, all of our coherence values, and then we would interpret that as all cortical signal with no interference from anything else. So traditionally, even though we know that the long separation channel is actually signal from the scalp and also signal from the brain, we just assume that the signal from the scalp is 0 and proceed with our statistics. But we can use something called a short separation channel to actually directly measure changes in hemodynamics in the scalp. So we don't have to assume this is 0, we can actually just measure what it is and then regress it out of our long separation channels for analysis. So a short separation channel operates by having a short separation between the source and detector. So our channel depth, that banana shape in the red, is just a bit shallower, and instead of actually measuring from the cortex, we're just measuring from the scalp. So it's a very simple concept. And then we can, as I said, regress that signal out of our signal of interest. Again, all of those scripts are on GitHub if you're interested. So our first question was, what happens to our coherence values when we actually reduce the signal from the scalp? Do we see that it has any effect at all on our average measures of coherence between children and parents, or does it seem like it actually doesn't really make that big of a difference? In oxygenated hemoglobin, we found that our regression significantly reduced our average value of coherence in every condition. So you can see here the dark red is our raw data with no regression, and then our lighter red is with our superficial signal regression per condition. And for each one, we see significantly higher average coherence in raw data than in regressed data. And in deoxy, we see the same thing in our 2 collaboration conditions, but not in our individual condition, although you can see, even though it might not be statistically significant, it does seem that the average is lower in our regressed data. So it seems like our regression is having an impact. It's reducing overall or dampening our average coherence strength across our conditions in both oxy and deoxy. So the next question then is, despite that, do we still see the condition effects that we would expect to see? So do we still see that coherence during full collaboration is stronger than during individual problem solving? That initial kind of core question of most collaborative hyperscanning studies. And at the whole brain level, we do see that synchrony is stronger in collaboration than individual in both oxy and deoxy, which I think make it an even more compelling result. And in this bigger figure here, you can see all of the channel-wise comparisons that survived FDR correction for multiple comparisons, where synchrony was stronger in collaboration than individual. And at the top, you see oxy, and the bottom, you see deoxy. The next question is, do we see that synchrony is stronger when I can look to the other person? So I might be able to see where they're looking. I might kind of get information about what they're saying from their lips or what they're feeling from their facial expressions. Or do we see that synchrony emerges even when I can't get that information from your face? So this allows us to test basically, is face information necessary for synchrony to emerge or not? And at the whole brain level, we don't see differences between the collaboration and partial collaboration conditions in either oxy or deoxy. There's a handful of channel-wise comparisons that survive FDR correction in both, but it's not a really robust finding. But of course, we would think that if you are collaborating at all, even if you're not able to see each other and you might be looking at each other's hands or talking and working on the same thing, we would expect synchrony in that condition to be stronger than if you're just working by yourself. So this is sort of a sanity check that actually any amount of collaboration or collaborative interaction, regardless of whether you can look to the person's face, is still kind of leading to the emergence of neural synchrony. And we do see that. So average coherence in collaboration with the screen is still stronger than individual problem solving, again, in both oxy and deoxy. And you can see the channel-wise pattern again here. Of course, all of this could be that synchrony is, it could just all be because we're in the same common task and environment. So it could be that synchrony is just an epiphenomenon. I'm sure that others have touched on this in this course, and it's a really difficult but important question. It's particularly hard to address in the context of naturalistic interaction because wavelet transform coherence relies on 2 signals of identical length, as I'm sure all of you know. So you can't very easily randomly pair child 1 and parent 10 and then calculate coherence between them. You can do that, but there are some drawbacks. You would need to splice all of your conditions and then feed each individual condition into your wavelet transform calculation, which means that they are much shorter. So you would feed in maybe 3 2-minute conditions for a dyad. What that does is it actually reduces your the number of wavelets that you can fit into that calculation. So it reduces your granularity at low frequencies especially. So you're losing out on resolution just so that you can kind of force these two random pairs together. And then, of course, you have to compare them to true dyads that have been done the same way. So it creates this problem where you're kind of making a trade-off just because of the structure of the wavelet transform coherence calculation. So we tried something a bit different called phase scrambling approach. What that does, well, actually, first, just to say again, in a true dyad analysis, we take our mother's signal, child signal, calculate our wavelet transform coherence, average across our frequency band of interest, and carry on with our statistics. But what a phase scrambling approach allows us to do is take the mother's signal from the true dyad to randomize the temporal structure of the signal. We did this in 100 iterations, which means we scrambled that signal 100 times. This maintains the signal amplitude, which is thought to be likely related to environment and task-related aspects. It also maintains the overall experimental time course, so we're not changing over the whole time series anything about kind of how short or long it is. We're just changing the temporal structure of the signal. We can then average across our 100 iterations, which gives us a null distribution, and average across our frequency band of interest and carry on. So this allows us to test if synchrony is driven by, at least partially, by the temporal dynamics of an interaction. If so, we would expect our true dyads to show stronger coherence than our pseudo-dyads. If not, if it's actually just that we're in the same place doing the same thing, and that information is preserved in the pseudo-dyad signal, then we might expect them not to be different. But what we found in both oxy and deoxy is that our true dyads showed stronger coherence in collaboration than our pseudo-dyads, and that there were no differences in individual conditions. So taken together, I think all of this suggests a few important things. The first is that I think that there's a potential influence of systemic physiology from the extracerebral layer that might actually be inflating our measures of neural synchrony, particularly during naturalistic studies, and particularly in oxygenated hemoglobin, which we know is more susceptible to physiological contamination anyway than deoxy. So I think it's important that we think about, when we're designing naturalistic studies, how will we address this issue so that we're not artificially inflating our measurements. We also see that this finding of neural coherence emerging during collaborative problem solving compared to individual problem solving seems to be robust. So we were able to replicate that even after reducing the impact of physiological contamination. So that's not the only reason that we might see that difference, that effect. We also found coherence can emerge in the absence of facial information, perhaps just as strongly as when face information is available. Of course, this doesn't mean that eye gaze isn't important. Of course it is, but it's likely not necessary. And so I think there's more work to be done about sort of what the role of that coordinated eye gaze truly is in the emergence of neural synchrony. And I think our pseudo-dyad analysis suggests that coherence is not entirely explained by shared perceptual processing. What this study doesn't tell us is why synchrony might emerge. And I think that this is a question that we don't have the answer to, a clear single answer to at the moment. If anyone does, please send me an email. But I think that this is something we need to continue to talk about and try to get to the bottom of in our hyperscanning research, so that we know we have strong theoretical models for why this might emerge, potentially what the functional role of synchrony might be, particularly in children's learning, in my case, and that we can kind of refine our theoretical models of what this actually is. There's lots of non-mutually exclusive explanations. They likely all contribute to the emergence of synchrony during collaboration. There's lots of discussion about joint planning, motor action, coordination of movement, language and communication, lots of recent interest, especially that I'm sure you've heard about, in the role of bond or attachment to your partner. And there's also this point of mutual prediction. So mutual prediction theory is one possible explanation that suggests that I, as an interacting human, have neurons that encode my own behavior and also that encode my predictions of other people's behaviors. And if I'm interacting with someone in the audience who's an adult that has the same neural architecture as I do, that the sum of our brain activity would be similar. So if we're doing similar things, it would be similar in that way, and therefore be correlated in our neural synchrony measurements. And there is evidence, of course, from single cell recordings in mice that this is likely part of an explanation, but of course we can't do single cell recordings in human children. And there's also really interesting stimulation studies that suggest that it could be causal. If we can cause neural synchrony to increase, that we might improve mutual prediction, but again, we can't do that with children. What we can do is build models called cross-brain GLMs, where we're modeling the activity of the child's brain in terms of their own behavior, their parent's behavior, and the brain activity of the parent. And the output of these models allow us to measure the relative contribution of each different factor within the model, which is a lot easier to interpret than the results of study 1 I've just showed you, where we don't really have that level of information. So we are in the midst of coding coordinated eye gaze, so the degree to which the parent and child look to each other, the puzzles and the templates, the extent to which they work together. At the moment, we're thinking about coding for instances where one of the hands of each person is touching the pieces at the same time, so they're working together in collaboration, and parent scaffolding behaviors, so how much the parent is kind of helping the child to complete the task. Most of this has been coded behaviorally so far by our master's student last year, Natalie Urbenska, using ELAN. This is showing you what the worldview of each eye tracker looks like. So the parent, the one where the parent is in, is the child's eye tracker, and then the opposite for the parents. And we can code instances of looking to each other, looking to the puzzle, parent positioning a piece or handing the piece to the child, those kinds of different things. But we also might imagine that that's not actually as precise as we want. Maybe we want something that's a bit more objective. So we're working with Ori Azmi, who's an expert in motor development and an engineer by training, to use computer vision to track dynamic areas of interest and also movement. So we can track, for example, parents pointing, child's pointing, as you're seeing there. And we can potentially measure different elements of collaboration that way as well. So this is just another visual way of looking at this. We're aiming to build models with the outcome of the child's fNIRS signal, and then add regressors for coordinated eye gaze, degree of working together, parent scaffolding behavior, and brain signal of the mother. And so this allows us to test whether the child's brain activity is modulated by each of those individual factors. But of course, there's lots of other ways to get to the bottom of what synchrony is. So this can tell us about maybe the really kind of granular dynamics of a certain type of interaction under certain conditions. But we could also test the emergence of synchrony in lots of different contexts. So you may have seen this already. This is a really fascinating plot that I think summarizes quite well where we might go with hyperscanning. We have two different axes here. So we have the x-axis where we have interactivity. We could imagine hyperscanning during co-watching. That's a popular thing to do, where we're not actually interacting with other people, but we're just watching the same thing. Or we could imagine a really interactive setting, which is kind of the area that I'm talking about, where we're cooperating, we're working together, we're having a conversation, and there's lots of interesting questions to ask about along that axis. There's also this y-axis of interpersonal closeness. So we might not know the people we're interacting with, or they might be, our partner or our parents or our friends, where we know them really well. And all of this work so far that's referenced here on this plot is done with adults. So we don't know what any of this really looks like in children. And our next study is aiming to modulate this y-axis of interpersonal closeness to try to understand whether familiarity of your partner plays a role in modulating neural synchrony between two children during interaction. So I want to kind of finish by talking about our work in progress, our data collection progress on this study about the role of familiarity. So we're collecting data from strangers, friends, and siblings who are either 3 or 5, the pairs of kids. And this will allow us to test both the role of age. So do we see that as collaborative skills develop from 3 to 5, that we see stronger synchrony in the older than younger dyads, and also, of course, to test the role of familiarity. We need about 120 pairs of child dyads in order to address that question, and we have collected about 66 so far, so we are well in the middle of this effort. And as you can see, it goes in these parentheses here, we have retention per group. So this is the proportion of good dyads that we get out of our testing. And as you can see, very often it goes really well. But occasionally, particularly in the case of 3-year-old stranger dyads, it's much more challenging. So I want to finish by just talking about some of our challenges in collecting this dual child data. If anyone is interested in doing child-child hyperscanning with fNIRS, please send me an email. Or if you have any particularly effective tips, I'm also very open to hearing about them. It's a really challenging thing to do, but I'll just kind of tell you what we've learned so far. Our first challenge is really getting the equipment on. So in this video, I'm putting the backpack on the child. This is a 3-year-old interacting with a stranger. You can see he's really not happy about wearing the backpack. So I'm asking him questions like, would you like to watch Paw Patrol? Would you like some stickers? Would you like to blow bubbles? Do you not want to wear the backpack? And so there's lots of bargaining that goes on, even not with the fNIRS system, but with like anything else that we want to put on them. Other times we get the caps on perfectly easily and it's great. The kids are happy with us putting them on, but they don't stay on for that long. So we do lots of catching the equipment. So she was happy to wear it initially, but then not so much. And sometimes we get really far. So in this case, the parent is kind of sitting in the position where the child needs to be. She's wearing her own cap without optodes. We're getting ready to go, and the parent is about to make the critical move to put the child on the chair so that we can start the paradigm. And of course, it's not always as we expect. So things that I've learned help us with this are stickers, bargaining, genuinely explaining what the equipment is. So we say things like, you're going to wear this cap, it measures your brain activity, kind of using like a Christmas tree. We're going to light up your brain using these little tiny lights, and it's really cool, and you'll get to see your heart rate in your brain signal afterwards. And we show them the OxySoft screen. And even with little ones who are 3, just 3, I think that really helps. They're curious, they want to know why they're there doing all this stuff, and actually explaining it has really helped us. We also only ever ask this or that questions. We don't ask, do you want to wear the cap? We ask, do you want to wear the cap now, or do you want a sticker first? So that kind of structure really helps. Despite those videos, we do often get really fantastic data, and it's been such a joy to watch this work well. I've never seen anything like it scientifically, where you have siblings kind of supporting each other, saying, well done, you did it, or kind of encouraging each other in a really lovely way. So I think we're going to get some really interesting data out of this design. And they often have a great time and are really excited and proud that they participated. Just to prove that it can work. The audio is a bit low, but they're just talking about this goes there, okay, right, okay, and then they'll correct each other and say good job and have, you know, a good experience. So overall, about 85% of our dyads do wear the caps. When they don't, a 3-year-old stranger dyad is generally the cause. So I would say that for the purpose of child-child hyperscanning, the 5-year-olds have no problem. The 3-year-olds who are participating with parents or siblings, well, actually not parents, that was 4 to 6-year-olds, but either in a parent's child dyad or a 3-year-old with a sibling who's 4, 5, or 6, they do really well. It's really just when they don't know each other that we've had a bit of a struggle, but it's still very possible, and we get good data more than half the time. Finishing the task can be challenging. We end up with children under the table or doing something silly. We deal with that by just adding flexibility where we can. So we'll have our 2-minute tasks that are not super flexible, where we really have to get through it, but then they might, between their task and their rest, have a moment of being silly if we absolutely have to do that, which sometimes we do. We also ask if they need loo breaks ahead of time, and we make it really fun. So we say, you're going to blast off into space wearing your goggles and your hat and your space pack, and we give them lots of space-themed toys and stickers. And then finally, we might imagine looking at all of this, thinking, well, the artifacts must be really difficult to handle. You have kids doing all kinds of silly things. But actually, our data quality, when they wear the caps, our data quality has been better in this study than in our parent-child study. I think just as a measure or factor of hair. So we see often really great coupling. We know that we're measuring hemodynamic activity with this really clear heart rate. And again, lots of the parents take pictures of this in OxySoft and bring it in. We've had pictures of kids presenting it to their show and tell in their classes and things. And kind of telling all their friends that they've participated in science. Recently, someone said that they've actually framed their little certificates that we give them. So I think it's actually been quite a positive experience from my perspective, looking at the data, as well as from the children's perspective. I'm running out of time, so I'm just going to say briefly that when we struggle with data, it's often actually the prefrontal channels. And this is, I think, just because the kids grab them first. So they feel them more, they're on their foreheads, they're itchier, they might be sweatier, so they tend to grab or touch the front. But despite that, we get really great data quality. In the future, we have, I'll end by saying, I'll try not to rush, even though I think I'm out of time, but we have lots of other hyperscanning research happening in the lab that's kind of all part of this package of that I've talked about today in a few specific situations. Our next plan is actually a longitudinal study of a classroom. So we're going to have a qualified early years teacher into the lab every other Saturday for 3 months, and we can investigate in a group of children, 4 or 5 children we're thinking at the moment, the role of synchrony in learning over time. And we also plan to have more naturalistic collaborative tasks like cleaning up the room together or moving pieces around the lab. So we've kept it to the table for now, but we're planning to make that much more kind of like a typical classroom task. We also have 2 wonderful PhD students who are working on different questions. So Imogen Green is looking at neural synchrony between slightly older children who are 9 to 10 with their teacher. And she's modulating or she's manipulating teaching styles. So either more scaffolding step by step and letting the child engage with that, or a more explanatory one-way lecture. She's also manipulating visibility, so whether the teacher is visible or not to the student. And really interesting, she's also testing learning mode, so online versus in-person teaching. So they'll either be in separate rooms on Zoom, each wearing a cap, or they'll be in the lab together in an in-person kind of configuration. So if you're interested in those topics, keep an eye out for some of her work coming up, hopefully soon. And then finally, Becky Terry, who's working with 2 to 3-year-olds and their parents in very similar collaborative tasks. And she's recently started a study looking at synchrony between the parent and the child, while the parent's actually just watching their child interact with a different adult. So hopefully all of this gives you an overview of kind of what we're doing at the Toddler Lab to understand the development of social cognition and collaborative skills in childhood. We have a few examples of research with 4 to 6-year-olds or 2 to 3-year-olds with their parents, and also, of course, trying to integrate some of those multimodal signals with fNIRS information. At the moment, collecting data about how children interact from each other in friends, siblings, and strangers. And then in the future, and also with Imogen's study, looking at kind of classroom or teaching style effects of neural synchrony. Thank you so much for your attention today. I know it's probably warm where lots of you are, different times all over the world, so I appreciate you being here. And thank you, of course, to all of these people pictured and named for their help, without whom this work really wouldn't happen. And of course, all of our participants who are in the hundreds now at this point for bearing with us and wearing those caps. And yeah, I'm happy to answer any questions.